I'm Eun Jung Choi, in charge of the protein engineering project at Blossom Park, CJ's R&D center located in Suwon, Korea. Before I start my talk, I would like to give a quick introduction about Blossom Park, or BP for short, and the Enzyme Research Group, where the work pertaining to the main part of my talk was carried out. Following that, I will talk about CJ's new Xylanase product, Xylanase, its structure and function relationship, its biochemical characteristics, and other functionalities it possesses. This is the street view of BP. The building encases a world-class research environment within its boundaries, with approximately 1,000 researchers working in the field of green biotechnology, white biotechnology, food technology, and biopharmaceutical research. From the sky view, the building is shaped like a three-petal flower, and thus is named Blossom Park. The Enzyme Research Group is part of the Animal Nutrition and Health Division of the Biotechnology Research Institute. Animal Nutrition and Health Division is focused on developing feed additives, including amino acids and enzymes. The Enzyme Research Group is composed of 6 projects, each with its own function. There's the discovery, engineering, fungi strain development, bacillus strain development, assay, and process development project. And this organization allows many different disciplines to come together to achieve the same goal. The discovery project is where all enzyme groups' research begins. New items for novel product is explored and discovered. Once the target is determined, the engineering project jumps in to improve the enzyme for the particular function of interest. The improved enzyme is then delivered to the strain development projects to be developed into production strain. Fermentation and purification is optimized by the process development project, and the final product is evaluated by the assay project. Okay. So the main part of my talk will be a little different from the usual feed enzyme talk, which focuses mostly on in vitro test or animal trial results. I will talk about structural and biochemical characteristics of CJ's Xylanase, CJ's new and improved Xylanase for feed application. It is one of the many products in our new feed enzyme product line, Growing Act. Xylanase was developed by utilizing state-of-the-art enzyme technology at BP to reinforce a natural Xylanase originating from an anaerobic fungi which lives in the alimentary tract of herbivores. And I will come back later to why this origin of Xylanase is important for its high functionality. You might remember from your biochemistry class that Christian Andersen's fundamental principle of protein chemistry states that protein sequence defines the protein structure, which defines the protein function. Based on this, I will explain a couple structural characteristics of Xylanase and the consequential functionality resulting from it. Xylanase is a GH11 family xylanase with the signature beta jelly roll fold. It is shown as a cyan cartoon representation in this figure. Antiparallel beta strands bend to produce the substrate binding groove of the active site. The active site is composed of 2 glutamate residues, which is involved in acid-base catalyzed hydrolysis of the substrate, which is the xylose backbone represented as a blue xylopentose in this figure. You can see how the substrate is tightly encased by the xylanase, and that explains the high substrate affinity for this family of glycoside hydrolases. The small size and high substrate affinity of GH xylanases are optimal for various applications, including feed. Another functionality of Xylanase that can be explained by its structural characteristic is its insensitivity to xylanase inhibitors. Xylanase inhibitors are produced by plants to use as a defense mechanism against phytopathogenic microorganisms. Xylanase inhibitors in feed is thought to hamper the functionality of exogenous xylanase added to the feed, thus lowering the quality and increasing the variation in the feed end product. This blue cartoon representation is a crystal structure of a fungal xylanase from penicillium, which is sensitive to xylanase inhibitors. It gets inhibited. The crystal structure shows it being inhibited by interacting with XIP, a xylanase inhibitor found in wheat. XIP is in light blue surface representation in the figure. You can see a long loop of XIP projecting into the active site of the xylanase and blocking the substrate binding groove. The arginine at the tip of the loop makes extensive interaction with many residues in the binding site, including the catalytic glutamate, and this is what causes the nanomolar affinity of xylanase inhibitors for xylanases. As can be seen in the table to the right from a reference paper published in BBA in 2004, bacteria xylanases are not inhibited by xylanase inhibitors and fungal xylanases are. Courgette xylanase is a xylanase which originates from anaerobic fungi which lives in the rumen of herbivores. In the literature, there are evidences that support the theory of hydrolase genes from anaerobic fungi having a prokaryotic origin, specifically from bacteria which lives in the same rumen environment. Courgette xylanase is from an anaerobic fungi, which, as I just said, are thought to have originated from prokaryotes, and thus are insensitive to xylanase inhibitors like their bacterial counterpart. This insensitivity to xylanase inhibitors can be explained structurally as seen in this figure. This is the same structure of inhibitor-sensitive xylanase interacting with XIP that you saw on the prior slide. In addition to the arginine at the tip of the loop, you can see hairpin loops at either end of the binding groove, which also makes extensive contact with the inhibitor. When you superimpose the structure of Courgette xylanase in cyan with the inhibitor-sensitive xylanase in blue, you can see the structural difference of the hairpin loops. Because of this structural difference, Courgette xylanase loops makes major clashes with the inhibitor colored in red. And this is thought to cause the difference in inhibitor sensitivity between fungal xylanases, which are sensitive to inhibitors, and Courgette xylanase, which is not. This movie depicts what might happen when an inhibitor-sensitive xylanase comes into contact with an inhibitor. The inhibitor is able to make the necessary contacts with the enzyme. It binds to the xylanase and inhibits the enzyme. In contrast, the movie to the right might represent how the loop structure of Courgette xylanase blocks the critical arginine-containing loop of the inhibitor from projecting into the active site and prevent binding. Thus, Courgette xylanase is not affected by xylanase inhibitors. To confirm this structure and function relationship of Courgette xylanase experimentally, we did an inhibition assay using XIP and Taxi, two different types of xylanase inhibitors found in cereal. As you can see in the graph, Courgette xylanase in red was not inhibited by XIP nor by two different isomers of Taxi. In contrast, xylanase originating from Trichoderma rhizae in green was inhibited. Trichoderma rhizae is a very well-known aerobic fungi and is the origin of many xylanase products currently on the market. Next, I will talk about the biochemical characteristics of Courgette xylanase. Enzymes useful in industry needs to be stable in broad spectrum of pH and high temperatures. To create that type of robustness, you need to modify in a very specific way the evolvable capability of natural enzymes, and among the many modified candidates, identify the best one. We made additional performance improvement on our native xylanase, which already had a very high activity, and the inhibitor insensitive functionality explained in the previous slides by using these methods to acquire the new and improved Courgette xylanase. This is the temperature stability data of Courgette xylanase. As you can see, it withstands heat up to 90 degrees Celsius for 3 minutes. It retains 100% activity up to 70 degrees and over 80% activity at 80 and 85 degrees Celsius. It is thermostable intrinsically, thus no coating is required, providing fast-acting product that can withstand commercial pelleting processes. The thermodynamic stability of Corejet Xylanase was also analyzed by differential scanning calorimetry. The melting temperature was 87.2 degrees Celsius, significantly higher than many xylanases in the literature. We also looked at the pH profile of Corejet Xylanase. The broad pH profile shown in red shows optimal activity between pH 5 and 7.5, indicating that in the small intestine, where the pH environment is neutral and where feed spends the most time and is degraded and absorbed, optimal performance is expected. Corejet Xylanase also has impressive pH stability, where it can withstand up to 5 hours of incubation between pH 3 and 10, with no decrease in activity at both extreme acidic and extreme alkali conditions. The ability for it to withstand extreme acidic conditions will allow it to pass through the upper digestive system of the animal without any loss of activity. The final 2 slides of my talk will be on the functional characteristics of Corejet Xylanase, substrate specificity, and xylo-oligosaccharide hydrolysis pattern analysis. The specific activity of Corejet Xylanase has been improved from its native form, labeled as wild type, indifferent to the type of substrate used for the evaluation. Compared to xylanases from Trichoderma or the native form of the enzyme, high specific activity for various xylans from different sources is observed, whether xylan is from beechwood, bagasse, wheat, or birchwood. Finally, we looked at the xylo-oligosaccharide hydrolysis pattern of Corejet Xylanase with beechwood xylan and wheat xylan. Xylo-oligosaccharide is an emerging prebiotic, and Dr. Kim has already given a full talk on the positive effects of XOS on animal health, so I won't go into detail, but will only mention that Corejet Xylanase cleaves both beechwood and wheat xylan into low DP xylo-oligosaccharides, which are thought to promote growth of beneficial microbials in the intestine. It also does not produce high amount of xylose, which is thought to cause viscosity increase in the gut and reduce nutrition digestibility. When cleaving wheat xylan compared to a competitor's product, Corejet Xylanase produce more xylopentose and xylohexose, which is beneficial to gut health, and less xylose, which is not. So this is the end of my talk. I highlighted the structural, biochemical, and functional advantages of Corejet Xylanase, CJ's new and improved xylanase, which will get you the most performance out of the feed you're using. Other feed enzymes, including manganase, glucanase, proteases, and phytase, are currently in our development pipeline and being improved with our enzyme technology. So keep an eye out for our new feed enzyme products, which will be available to our customers soon. Thank you for listening.
Grow and Act.